So I would like to follow me to see what's going on in the sea bass and sea bream, okay, species that are more interesting for us in Greece. So I'm going to present you the application of genomic tools and techniques to sea bass and sea bream farming. And for those that do not know me, I'm part of the Hellenic Center for Marine Research in Heraklion, in Crete, and we have recently fused with the Institute of Aquaculture, so to form a new Institute of Marine Biology, Biotechnology and Aquaculture. So we're also part of the same institute with people that you might know, like Dinos Milonas, Papandoublakis, and so on. I think that Panos changed a lot of things in the presentations, and all the letters became gray instead of black, because he had to put his logo I don't know if he would agree to have my initial presentation, but nevertheless, okay, through this presentation I will try to discuss the principal, potential, power and advantages of different DNA markers to aquaculture species in order to assess genetic variability in breeding, outbreeding, to see how species and strains can be identified, also to see if it's going to be used for hybridization and parentage assignments, and last, the use of molecular markers to help in marker-assisted selection and the identification of QTL. I will try to explain all that, focused on the two species that we were talking about, sea bass and sea bream. The geneticians mostly would like to think that the development of DNA-based techniques have had a revolutionary impact on animal genetics, and with the DNA markers it is now possible to observe and exploit all the genetic variation of the genome. And also, if we perform well-designed studies using appropriate genetic markers, it is now possible to find and identify genes that are involved in the so-called quantitative trait loci. That means the genes, the regions of the DNA that are there to control a function, to control a reaction of the organism, and for the genetic improvement, having always that in the back of our head, how to improve all the stocks and the characteristics of the aquaculture production that has to do with efficiency, with the making the product quality increase, and of course that at the end will be the profitability for the aquaculture enterprises. So as I'm coming fourth in the row, so lots of the things have been presented by other people in the room. But okay, I'm glad to see that there are not so many duplicated slides. So in order to monitor all the genetic variation, we need to have polymorphic markers. So and markers that should be heritable. That means that it should be easily monitored from one generation to the other, and easy to be identified. At the DNA level, let's forget the old-fashioned techniques and markers like allozymes that have to do with squeezing tissues and getting the juices and enzymes out of that. We have now the sophisticated DNA techniques and markers like SNPs. So it's base substitutions. Instead of an adenine, it's a guanine, and so on. So we have just simple mutations in the genome we may have insertions and deletions. Insertions and deletions in the genome, inversions, and as other speakers were telling, this can be grouped into type 1 markers, markers that have to do with the function, and type 2 markers that are supposedly neutral, but are neutral because we don't know if they're neutral because we don't know yet if these markers can be associated with the gene. So that's why the type 1 markers are more sure, and the type 2 markers are maybe lots of them could be classified after some months or years when the genome will be more analyzed and annotated as class 1 markers, functional markers. If we go back only 8 years ago from publications from you and Costas in Aquaculture magazine, both were seeing in the future the replacement of techniques with other more sophisticated techniques like FLPs, microsatellites, SNPs, and they were saying that more and more laboratories would have the possibilities to buy one of these machines and do fancy for that period things. And it's true that as technology progresses, more and more labs, more and more companies are able to do more sophisticated and fancy things, which 20 years ago were just a dream. So you finally used the first or the second presentation that I sent you?
You missed slide.
Yes, so what type of marker to use? That's an eternal question to genetics, but that has a simple reply because that depends always on what type of questions we have to resolve. So for questions that have to do with phenomena that are more recent, we have to use markers that are changing a lot, so polymorphic markers. And when we go to the past, the type of markers to be used are different classes of markers. So in our case, talking about the identification of strains, of inbreeding, and so on, the most informative markers to use are microsatellite SNPs and techniques like the real-time PCR, the quantitative PCR, and the DNA microarrays. So, if we start with the genetic diversity and resource analysis, Panos, I think it's the first presentation that I sent you. Do you think it's possible to plug the other one? I have it if you like to put it directly.
Just a moment.
Do you mind?
No, I don't.
Sorry for that. Sure. But now it's true that we can see better now. Okay. In spite, so concerning the aquaculture stocks that we use, in spite of the relatively, when we say relatively, it's sometimes recent aquaculture history, broodstocks are not well characterized from a genetic point of view. Many strains and aquaculture species are used, but the genetic relationships among strains is often unknown. And there are questions that are popping up to see how diverse is the genetic background of the aquaculture broodstocks that we use, and how they differ from the wild counterparts or species or populations coming from different parts of the distribution of the species. Traditionally, okay, we have started with enzymes and mitochondrial DNA, and nowadays we use more sophisticated techniques like SNPs and microsatellites. So, for the European sea bass, what we know for now is that all the studies that have been performed up to now show that the sea bass populations can be grouped into three different stocks. We have the northeastern Atlantic Ocean stock, the Western Mediterranean, and the Eastern Mediterranean stocks. These are the three main stocks of the Atlantic of the common sea bass. Okay, when we say stocks, it's not, for example, the limit between the Atlantic and the Mediterranean is not exactly the Gibraltar Strait, but it's mostly the Almeria-Oran front, because we have to think not the idea of the geographic separation, but mostly because of the sea currents that circulate, and so the water up to Almeria-Oran is mostly of Atlantic, let's say, origin, because the circulation of the waters make these waters a little bit special, and the separation between the Western and the Eastern Mediterranean stocks could be positioned close to the strait between Sicily and Tunisia. And okay, at the beginning it was a little bit surprised, because sea bass is used to be considered as a urea-line and euthermic species, a species that can migrate a lot. So this differentiation, it was quite strange to see. So here are the points and the studies that are used, have been used up to now to do the population genetics of sea bass. So you can see that most of the regions have been sampled up to Egypt and northern France, in order to get this differentiation into three stocks. And for example, there were studies that showed that even along the Iberian Peninsula, there are different stocks, and recent studies that show that there is a recent migration of the sea bass, and we will see that afterwards also for sea bream, to the north, because it's the climate change, so the water is becoming warmer, and now we can find even Greek sea bream up to Ireland. Now it's reaching Ireland, because the water is becoming warmer. And here we could also see a study in the Eastern Mediterranean by Castillo and Cifci some 7 years ago, when we had samples from Patras, Hellenic, and Egypt, and they could differentiate all these populations. So even in the Eastern Mediterranean, we could, if we make a zoom, for example, we could see differentiation between different localities. Concerning now the gilted sea bream, Up to now, we use different type of markers, and all the studies show that there is a slight differentiation between the Mediterranean and the Atlantic, but not so striking as between S for sea bass. And in general, there is not an homogeneity, but there is no genetic structure when we take sea bream populations in the Mediterranean or in the Atlantic. And it appears also that in Asia or Northern Africa to have different kinds of gilted sea bream, but these results are quite recent, and more populations should be analyzed from Northern Africa before having definitive results. But it appears that North Africa has a separate type of populations. Okay, here we could see, compared to sea bass, the sea bream studies have lower samples of populations in the Mediterranean and in the Atlantic. And nevertheless, if we use lots of markers now, we have analyzed up to now 96 different microsatellites, and we can now go and make a zoom and differentiate population from the French coast, Portugal, Greece, and even between Greece and the Adriatic Sea. And that's also another example that I was telling to you earlier, of the northern expansion of the sea bream to the north, to see in fact where it comes from, if it comes from the Portuguese or the French waters, but it was not easy to make some solid conclusions. Okay. For species and strain and habitat identification, it's also very important to have an idea of the genetic structure in aquaculture, because fish stocks sometimes, as in tilapia, may be hybrids, and so sometimes we spend time and time investing in stocks that we think that are pure stocks or wild, and in fact we have bought something that it's not correct. So here again we have microsatellites in the FLPs that have been used, and as discussed earlier, is the use of allele frequencies analysis that may help us to see the differences between the different stocks and strains that have been used to form our nucleus or our breeding scheme. Because inbreeding is in fact a major cause of losing money, because as you know, inbreeding is just the fact to mate sieves, to mate close relatives, and when we do that, for example, in simple breeding schemes, when we take the bigger fish or the more resistant fish and we choose them for the next generation, the main problem is that in many cases we choose sibs, we choose brothers, we choose cousins, and so in the next generation we have problems because it's the inbreeding that pops up, that appears, and so it's the production that goes down afterwards. That is why inbreeding should be managed through a structured breeding program that allows a high degree so inbreeding should be managed in a structured breeding program that allows a high degree of relationship to exist between the individuals, but prevents mating between close relatives. Because in few words, there's no meaning to attempt sometimes in-house selection with few broodstock that we have decided that have all the criteria to proceed to the next generation, and take the risk to make seeds, and finally have, as Costas was saying, this loss of genetic variation through genetic drift, because we are going to lose everything after some generations. And what we also forget to mention is the outbreeding depression, because we have inbreeding but outbreeding, and it's something that appears when we are crossing different strains sometimes. And what happens is that every strain, every population, every type of population, through the evolution, the genes have a certain position, and when we sometimes we cross different strains, different populations, all these equilibria are inverted, and so that may also cause a breakdown of these complexes and also to lose production. So what happens sometimes is that we have the first generation that is quite good in production, so we say that's okay when we cross two different populations, but what in fact happens is that in the second and the third generation, the problems start appearing, because it's the segregation of the alleles of the genes that form the outbreeding depression. Okay, that's an issue that has to do with escapees. Bjorn already talked about that. That was a European project trying to trace escapees. Sometimes for the companies it's a delicate issue to speak about, but nevertheless it's a problem that nowadays there is some concern, and maybe in a few years I don't think that we're going to be as Norway, I mean, to trace back and find all these escapees in the wild. But for example, in this study there were two sampling sites close to Alicante and Mesologi. So what they did was, let's say stupidly, sample one population from the farm and one population from the open sea, and see if they could trace differences. So already I didn't, it was not me that did the sampling, but already you see something strange, and that's strange is probably because the farmed Greek population is not following the general pattern, because it appears to be of different origin. That's for sea bream, for example. And so we could see that easily here. So we have the Greece wild, Greece farm, Spain wild, Spain farm. You could see that in Spain, for example, what's in the cages and what's in the wild, it's the same. So probably they use for the cages breeders of the wild, or the wild has been polluted, let's say, from fish from the cages. In Greece, what we have inside is of an unknown origin that has nothing to do with the wild. And in some cases, we could identify few fish that are closer, let's say, to the farm population. So this might be an indication of escapees, but okay, we don't have all the elements to say that. In sea bass, again, who is different? It's the farm Greek population again. And it's more or less the same. So again, we may have sampled an Atlantic origin farm Greek population. So the wild has nothing to do. In Spain, it looks homogeneous. If we zoom in the Spanish population, we may also find some differences, but these differences are local, and again it appears that there is an admixture between farmed and wild population. But here you can see wild Greek and wild farm. Nothing to do, but as I told you, it was something that concerned just an aquaculture company. I mean, it's not the image of all Greece or of the Mesologi region. At least for the wild, this is an image of the wild, but the the parent Greek, it's just a company. This is also a quite interesting study through this project. What they did is to evaluate the egg production of gifted sea bream that was kept in cages. What was happening was that a company or a few companies, I don't know who is doing that, they're keeping the fish to be grouped, to be bigger in their nets. So they have fish that can reach more than 1.5 kilos of sea bream. And what they did was to fabricate an egg net, and they put it off the cage, and they could collect eggs and larvae. So what they saw, and after we confirmed that by genetics and also by dissections and doing analysis of products, is that the sea bream started having eggs in the cages, so releasing millions of eggs in the wild. So we have to be cautious in the future, when handling all these individuals and let them release their eggs, because as we saw in the previous case, if it's sea bream of Greek origin, okay, we could say that it's an enrichment of the wild environment by Greek sea bream, but if it's sea bream of another origin, we just pollute our Greek stocks with fish brought from other origins. Now for parentage assignments and reproductive contribution with microsatellites mostly, we use them in the last 10 years to monitor the parentage assignment and what we call sometimes spawning kinetics, who is contributing for the next generation. And of course, sometimes we wonder how many microsatellites, how many markers do we need, and the question is not always easy, because we need more markers to be used when we need to analyze more fish. And there's an increasing demand to use markers now, previously microsatellites, now SNPs, to make a proper stock identification, but also to proceed to breeding schemes. And we will see that, for example, that when we want to use a parentage assignment, as we saw earlier in selective breeding, it is extremely important to obtain high and reliable assignment rates. We have to be sure that the fish that we choose are coming from these parents, from these brooders, and not from others. Because sometimes it is better to invest some money and be sure of the fish that we choose for the next generation, for example, than to rely on fish of an origin that we don't know and afterwards lose money in a breeding stage. What we also know sometimes is that it's not all the fish that contribute equally when we put, let's say, 20 females and 60 males. It's not every male that contributes in a mass spawning event, but there are sometimes dominant individuals that contribute a lot, and others that are more frightened and are keeping back, and so they don't contribute a lot to the next generation. So that is why sometimes this phenomena reduce the selective breeding values and the selective breeding results that we could have. Not to forget that in most of the cases, the most dominant individuals are not the individuals that we're interested in to ameliorate, to increase our production. It's just the individuals that are more eager to participate in the reproduction. As we have seen in fish, the assignment rates, for example, depending on the markers to use, are more or less quite quite efficient, but in sea bass, sometimes we may have some ratios that are quite low, that we have seen better, for example, in other studies. In the common sea bass, what we know, for example, is that the sea bass produces 3 or 4 or 5 spawns during the annual reproductive season. So when in 2007, in a study with Dimitris Katsikis, Costas S, we analyzed the commercial broodstock of 103 brooders, 58 females and 45 males, and we analyzed approximately 1,200 fish. It appeared to be only one female that participated, was responsible for 95% of all these 1,200 fish, and 26 males, of which one had a major contribution. So we didn't expect that. We thought that every fish will participate in that, but it was not true. It was just more or less 27 fish, one female and 26 males. When we did that, some 3 years afterwards, we thought that we would find the solution and go beyond. But again, we were found with 11 large full SIP families. And when we genotyped 2,000 individuals, we find 2 females and less than 10 males that participated. Again, a problem. In Bream, it's not the same because the Bream female are asynchronous multiple batch spawners, and what does it mean? They have an almost daily cycle in a period spanning 3 or 4 months in which they produce a smaller quantity of eggs compared to sea bass. So when Cameron Brown tried to calculate the effective population size, that means the actual number of breeders that participate in the next generation, using always microsatellites, they find 80 out of 54 Bream broodstock, nearly 1/3 of the Breams that participate in the next generation. And also Spanish colleagues, and a recent study by Dimitris Lokovitis in which me and Costas also participated, they also proved that this is more or less a standard ratio. 1/3 to half of them, nearly half of them participate in the next generation. And so the families that we finally have, it's not exactly the model of 5 to 5, so let's say 25 families, but in fact the families that we have are much less. So for DNA markers and QTL analysis and marker-assisted selection, where are we now? Lots of the people before me have already mentioned lots of the progress that has been made in the QTL analysis in sea bass and Bream. What we're interested in, and I'm going to show you, and you also saw that in Professor Canaris and Costas Batargia's presentations, were the fact that we need markers evenly spaced in order to cover as much as possible the genome of sea bass and Bream, to do what we call a genome scan and see and be more precise in the selection of the markers that are associated with the trait. So, for example, in the common sea bass, what we have up to now, as our colleague was presenting for salmon, we more or less have the same. We're not far behind from salmon. It's a mistake to consider that salmon, okay, it's much beyond. The production, it's another thing. But we also have a linkage map. We have an arrange map that they don't have, and a back map. And I'm going to present you in a few details. So we have a linkage map, but it's compared to salmon, it's 2 or 3 times less dense than that of salmon. but there is one. So all these groups, and if we could say its linkage group is one chromosome, let's make the comparison to that. Let me remind you that Bream and CBAS have 24 pairs of chromosomes. So 48 chromosomes, whereas salmon has more than 54, so some more chromosomes. And that's the coverage that we managed to have, 3.65 centimorgans. Some years ago, they managed to have an radiation hybrid map, which is simply they cut in pieces the genome of the sea bass and they put it in cells of hamster. And so they know which parts of the chromosome they are in the cells, and they can sequence that. And so they are able to see the close regions of the genome because they are all in the same cell of the hamster. And that's a great value afterwards when we are going to proceed to the next generation techniques. Sorry. When we put together the radiation hybrid and the linkage maps, we can see that there's a big overlap, but that also helps us correct few problems. This is an image to show you where is Bream and sea bass compared to the other model fish species. When we say model fish species, is the fugu and tetraodon, is the three-spined stickleback, is the medaka, and the zebrafish. And you can see where are the others. For example, the salmon trout. You can see that's huge, the difference between them. These are the colds, mallets, and so on. So when we are doing comparative mapping, we have interest to do that with close species and not to go very far in the evolution of the fish. Here, this is another example of the comparative radiation hybrid map in sea bass. We similarly constructed the back library that is already publicly available. And what happened afterwards was that we used the back-end sequencing through the Marine Genomics Europe Network of Excellence and the Max Planck Institute in Berlin to do a first step to the genome sequencing of the sea bass. And approximately 100,000 back-end sequences were aligned to the stickleback genome, which is a genome well annotated. And what finally happened is to cover 87% of the stickleback genome with sea bass sequences. Adelino told us previously that this rate is much bigger now because more sequences were added, and there was a better annotation. So we have this picture where in green are the sea bass sequences that have a match in the stickleback genome, and the red are the gaps. But again, the stickleback has 21 chromosomes, sea bass and Bream have 24. So there are some groups of stickleback that correspond to more than one chromosome for sea bass and Bream. But it's a big progress. And through this project, what we had more than 20,000 SNPs that were identified, and that's something huge. But sea bass, it's not a species with very few resources. Now there are lots of resources that have been developed for sea bass, and we are able now to proceed and construct microarrays. And we can see, for example, in the linkage group 1, let's say the first chromosome in sea bass, the distribution of the different SNPs in the length of the chromosome of the linkage group. So we have regions of less SNPs and other regions with more SNPs. So we're able to do, for example, selection on certain traits, knowing lots of informative markers in different positions of the genome of CBAS. So for Bream, as also for CBAS, there were some pioneering European projects that funded the generation of lots of resources, genomic resources, like BridgeMap, which was a project that was led by HCMR in Crete, and Marine Genomics Europe, in which Adelino Canario was the head at the fish node of the consortium, because Marine Genomics Europe was not only for fish, but it was for many different marine organisms and aqua first. So again, there is a back library. There was a third generation linkage map and an RH map, and also quantitative trait loci that were reported. And through the Marine Genomics Europe pipeline, there was a lot of non-redundant ESTs and contigs that were identified in different Bream tissues to help us also find SNPs and microsatellites. Here is the map of the Bream when we compare that to the tetraodon. This is the more recent version of the linkage map for Bream. I believe we'll have to publish that by the end of the year. I think otherwise it's going to be obsolete next year. That will help us transfer information, as we said before, the comparative mapping, into two directions to map genes that have not been mapped in other species, but also help us sort out markers that have not been mapped in Bream. But what is interesting also is that little by little we started also sequencing the Bream genome. And what happened up to now, it's not, of course, the 100,000 back-end SNPs, back-end sequences that were mapped in CBAS, but nevertheless, we have more or less like 15,000 clones and more than 5,000 backs that have started to be positioned on the Bream, on the stickleback genome. So, as previously for the CBAS, here in the first lane is the CBAS, here is the Bream, and here is the stickleback genome. So we have, as you see, a quite nice homology. That means that the two species are quite close to each other, and there is a synteny, how we call that. So what it can help us identify regions of interest in both species if we can transfer also information between model fish species and CBAS and Bream. For example, if we position a QTL of major importance in CBAS, we could also have the information on the same region for Bream and vice versa. So there is a continuous flux of information. Now, screening for candidate genes. Nowadays, we have high-throughput differential gene expression projects in CBAS and Bream. For example, in developmental stages, we have the cDNA macroarray. Infections with Vibrio and nodavirus. There are at least two publications that refer to nodavirus and Vibrio from a colleague in Crete, Elena Saropoulou, by using cDNA sequencing or next generation sequencing transcriptome, jaw deformities, fast skeletal muscle in the Bream with the Spanish team, Garcia de la Serrana Castillo. I think they are from Torre de la Sal lab, and also the animal welfare that has to do with stress. And so we have cDNA microarrays that are in preparation in CBAS, And the cDNA microarrays that have already been developed from Caldush Guinea, the French, the Spanish colleagues, and taking a few minutes to present you what has been done in CBAS from the last studies. There was a development of an oligo DNA microarray. A microarray was a chip like those presented earlier by Bjorn and also Adelino. We spot all the regions, all the DNA segments that we have, and we have probes of around 64, 65, 100 base pairs maximum, spotted on small metal plates, and we put our RNA from the tissues on these, and we can see if the probes are working. So if they are flashing red, for example, that means that they are working a lot. If they are flashing green, that means that this gene is not well responding to this signal. So, for example, they use an oligo DNA macroarray for CBAS to see the gene expression in mandibles and cold heads of fish affected by prognathism. You may have that in your aquaculture facilities, skeletal malformation that affects the CBAS production. So they use the Agilent SureSprint microarray technology to spot nearly, let's say, 20,000 target clusters. So let's say 20,000 genes. And so they identify 200 genes that are related with this malfunction, these jaw deformities in CBAS. And when they did the annotation of these genes, there were genes of the nervous development. So they know that there is a bone nervous system development that plays a role in the jaw deformities. Elena Saropoulou, a couple of months ago, when she published that, it was the characterization of CBAS transcripts by RNA sequencing. It was after oral vaccine against Vibrio. So they used two different tissues of gut and head kidney and the 454 next generation sequencing to generate some thousands of sequences. And so they saw and evaluated the efficacy of the vaccine, which revealed specific immune gene expression profiles in the gut and in the head kidney. So we have different genes that function when we give this vaccine, and other genes that do not function. Similarly, for Bream, they use this technology to identify sequences that have a key role in larval development, in differentiation in Bream. And a similar study in 2012 against infection by Pteromyxum. They saw what was the difference between fish that were fed with different fish oil diets, and when also these two types of fish were exposed to strains of Pteromyxum. So they saw that the high replacement of fish oil that does not change a lot the function of the intestine transcriptome, but what they saw were changes that were different in fish fed with oils and fish fed with vegetable oils. So for QTLs, we have to identify threats of interest and afterwards do either random scan for QTLs or targeted QTL scan when we have an idea of the region of the genome that we want to investigate, and to identify candidate regions or markers linked to the trait of interest in the worst case. So again, for CBAS, what has been done is up to now, Hatziplis identified a significant QTL for 6 morphometric traits, including length and depth, that was identified in 2007. Afterwards, there was a study in 2010 with our colleagues in Roslin University in a study in which we all participated, again to see the other two QTLs, but unfortunately it was not the same. But here we also put some more characteristics like the body weight and the stress response. So there are clues, there are ideas behind. We have indications for QTLs in the sea bass. So this is the picture that you already saw a couple of times with this marker in the first linkage group of sea bass and this QTL analysis that identified that, and also other QTLs for growth and body shape also in the sea bass. So we have the basis now. We could go a step beyond and not start from scratch. We're not at 0 point. We have some ideas. We have some markers. And we're open to collaborations to test them in your populations if you like. For Bream, there are at least 3 or 4 studies that have studied QTL analysis in hub SIB and variance components and identified a single unlinked marker, unfortunately explaining lots of the variation, and several other putative QTL analysis for other morphometric stress and also for stress response. So that was the idea of tomorrow, try to treat fish better and make their life as we say, okay, try to make it better. And recently Lukovitis identified 2 significant QTLs for body weight and one that is close to a region that may affect sex and sex determination in the sea breed. And they found them quite close, which is sometimes a general trend in sea bream, because sea bream, you know that very well, it's a protandrous fish. It's male. It is a male first, and afterwards it changes as female. And it appears that as the fish grows faster, it will change faster. And sometimes that has not to do with the period all the time, but to the tempo, to the rate of growth. So that's normal that the 2 QTLs are found close together, one for growth and one for sex reversal. And in a publication that, in a paper that's now to be published, there are also some 4 growth-related QTLs and 6 other QTLs that are proposed and can be used in the near future for marker-assisted selection. So these are the results from Boulton et al. On the QTLs they have found on body weight, and the others from Lukovitis. What is also interesting is to use the comparative mapping information, and what is strange also is that when we talk, for example, for the Asian sea bass that supposedly belongs to the same family with the European sea bass, in fact, the Asian sea bass is like sea bream. It's a fish that changes sex. So it's interesting to use sea bass, sea bream, and the Asian sea bass, which is sharing lots of characters in common with sea bream because it changes also sex. Whereas sea bass, okay, it's a monopolistic species, and so having an idea of the gilt-head sea bream gradation habit map and the position of QTLs on the map, we were able also to see the linkage group 1 of tilapia, which is also a very species which is very well advanced from a genomics point of view, and propose the regions that could be interesting for the sex determination in C3. So that's the QTLs, that's the next generation sequencing. What's the future? So what we think, okay, I like to present myself as a scientist, but as Bjorn was saying, science is fine. We like doing science, but it has to have an application side of science. So I don't think that science is only to keep it for scientists, but we have to share it and we have to communicate what we find to you people of aquaculture. And if you think, and if we can persuade you that it is important to invest time and money in your personal, I don't know, because sometimes it's not only a question of money, but time. You're also quite occupied, you have lots of things in your mind. You're not, you don't like to get puzzled with things that you cannot understand, but in that case, do not hesitate, just ask. And what we expect in the future is to increase the application of the more recent and emerging technologies to accelerate things in the aquaculture industry, to enhance production, and because the new markers are there, but they are also going to arrive with the application of the techniques. But we have to use them for you. It's interesting from evolutionary point of view to use them for science, but it's more interesting to use them for application purposes. And what we have now is the three major next generation sequences, is that we may have machines that are more or less the size of a laser printer. We used to have machines that were occupying, I don't know, 4 cubic meters, huge machines. And now the benchtop machines that you can have them next to you. And when I say you can have them, it's not like that, because as you saw, that costs a lot of money. But nowadays, sometimes it's not worthwhile investing. If you can tell me, if you had 600,000 euros, would you like to buy this machine? No. Someone else has the machines, and we could do the work elsewhere, and we could analyze the data together. It's pointless for a company to buy a machine. Now, for example, for us, even in the public domain, it's pointless to invest money and buy these machines. There are SMEs, there are companies that have them. They can sell the knowledge. We can share the data and analyze them together and put them together concerning your problems. If it's growth, if it's disease resistance, if it's whatever. And this is the amount of sequences that is generated in the last only 10 years. You can see that it's a log. It's a log phase. Every year, the amount of sequences that is generated for different species, it's huge. You cannot even imagine. We had in the last week a meeting in Crete, where I don't have the slides. It would be nice if you could put all this data in paper. You could probably reach the moon. You know, printing all this data and filling them, piling them up. You could reach, you know, it's thousands of millions of data. As Adelina was saying, the problem now is how to store them and how to analyze it. Sometimes you need big bioinformatic units, big clusters of computers to run them all together. For example, if you have results from a RAD tag sequencing, that means tag each individual separately. So in that case, you could analyze, for example, let's say 100 fish that were dead after the infection, and 100 fish that survived. So you say what's interesting to see in their genome. So you can do that, pulling all the individuals together and do them sequencing. And you can have idea on their on a big part of their genome, let's say 20–30%. But that's enough to have the first idea. But that generates lots of data that even if you need 1 week to run the sequencer, you may need 6 months to analyze the data afterwards, because you have information for lots of PhDs sometimes. And well, at the moment, where are we now is that we may generate lots of data with, for example, 2,000 to 3,000 euros. Okay, that's the cost of consumables. Afterwards, you may need personnel to analyze all this data, because if I can tell you, okay, take this book and you will learn about that, that's nothing. You have to be behind the people that are going to help you and work with them to understand all this evolution. Not to mention that, okay, we may have thousands and thousands of novels, projects. So, interesting things, for example, for CBAS and CBRAIN. Excuse me, I'm going to present you 3 projects that I know. There are probably others that you may know, Costas or Professor Canari or Joan. This is a project that I'm leading in HCMR that has to do with the transfer of knowledge from Bream to other spirits that could be potentially future species for aquaculture. And for that purpose, we are using the Bream, the common Bream, Pagus, the snapshot Bream, Glottis punctatio, the common Pandora, Phageolus erythrinus, and the common Dentex. So I forgot to mention that, as we saw, the CBAS genome is more or less close to its first draft. They are close to the first draft of the genome. Of course, when let's say 10 years ago, Craig Venter reported the first genome of the human, he was happy. But now what we see is that it's now maybe 100 times cheaper and 10 times, not 10 times, 100 times even faster to have resequencing of the human genome with the progress and all the technologies that have been invented ever since. So what's interesting, for example, for human genome, since for pharmacogenomics and for individual screening, is, for example, we have a problem, I have a problem, I give DNA, and they are able to sequence my genome in a week and propose to me therapies that are just for me. So the progress of the technology has achieved the point in which we may have these species ready to be used as model species for spirits. I have also to mention that the Bream genome is at a good point, but it's going to be to change dramatically maybe in the forthcoming months, because we have sent tissues to the Chinese, and they're going to do the full genome of the Bream in a year or so. So it will also be available. And when we have all this information from other close species, because what is interesting in the other spirits, for example, is that you have species that are gonochoristic, like Dentex. You have species that are protogenous, like, if I remember well, the Pagus and the Phageolus. So what if we are able, for example, to modify or to trace the genes that are responsible for sex determination? So fish that will mature quicker, fish that will not reverse their sexes, and so on. And also to speak about the project that is called Aquatrace. It's a project that is under negotiation yet in the European Union. With Panos, unfortunately, we're participating in a component consortium, which finally failed because they submitted their proposal 5 minutes delay in Brussels.
No, I remember.
You still remember? I'm trying to forget. And this project aims to monitor the changes in the not only in the domestication process, but also in the wild environment when the fish are released, either by intention or by accident. So they're going to develop lots of high throughput tools to monitor the escapees in the wild. So what Bjorn was describing in salmon and in Norway is going to happen in Bream, sea bass, turbot, salmon in a few years now. They will be able to, I don't know if they will do that, but if they will be at least in condition to use a chip and trace the escapees in the wild and say, this is the fish that has escaped from this farm or another farm. And the last one, this is a new call that has a deadline by the end of January, and it is supposed to boost the domestication of established finfish species through selective breeding. I don't know if Europe will give more money in sea bass and sea bream in the near future, but at least this is a project for the next 4 or 5 years. So you have sea bass and sea bream, but also salmon, rainbow trout, and turbot. So that's the interesting thing, is that we will investigate the possibilities for measurable, quantifiable genetic improvement by focusing in priority on some of the following traits resistant to pathogens and diseases that hamper the production and for which no efficient vaccine or no cost-efficient prevention treatment method exists. To my information, they will not insist so much to no efficient vaccine, so they will try to do efficient breeding schemes for every disease that causes lots of problems. So that's why your view on the disease that hampers production is quite important, because we cannot, for example, let's say, enter to a consortium like that without knowing what is your problem. If the French are coming and saying, I'm going to do Nota virus because we know that Nota virus is a big problem in France and Spain, and you say, that's interesting, I would also like to do Nota, and he says, you don't have problems of Nota virus in Greece. Because that's the statistics. If you don't say that, they consider that there is no problem of Nota virus in Greece.
Nowhere you can find statistics for Nota virus, nowhere in the Mediterranean.
I don't know, but when you talk with these people, they say, okay, we know that for us, at least in France and in Spain, Nota virus is a problem, and you may, for example, work with others like Vibrio or Spicotle or GENOMICS or others. So there will be also an effort to enhance growth in filleting yield in the species that we can. I don't know if sea bream can be.
Are you putting any proposal for this call with Avelina? Are you applying for this?
Not yet.
What's the deadline for that?
End of January.
Okay.
And the other one is adaptability to alternative feeds and changing diets. I mean, how to pick up families and strains that could be adapted better to different feeding conditions. So I would like to thank all my colleagues from Crete that helped me with this presentation. George Kotoulas and Ella Sarapoulou, compagnon d'armes of the past, like Dimitris and Costas, people with whom we have participated a lot in different projects, Adelino and Deborah, Luca and Tommaso, Philip, Brendan, Angelo, unfortunately he's not at life, and other people through which we participate in different parts of projects like Bridge BAP, Plasma Backup, First Prevent Escape, and Marine Genomics Europe. Thank you all for your attention.
Thank you, Costas. Any questions for Costas? I felt after the Norwegian presentation that we were light years behind, but you narrowed the distance. We are probably, I don't know.
You know, everybody has problems. The problem, I think, with salmon was the fact that you had different. What Bjorn was describing was the big fight between the Norwegians and the Canadians and the other people. So you have the same objectives into two teams in, let's say, in Norway, where you had, as you saw, lots of projects because they have money to put in. So you have national funding for small projects. On the other hand, of the Atlantic, you have the Canadians that were trying to compete the Norwegians and, in fact, produce twice the same data. So sometimes, okay, it's true that when you collaborate, you can make things go faster than to compete each one from each side. As Bjorn was saying, okay, much of the salmon production is not due to genomics yet, but we're at the point now, at the threshold where we have to think very, very seriously the use of genomics because, and they have seen that in salmon, they have reached a plateau. You cannot go beyond with the typical selection. And now you have to go into genomics, and instead of choosing families against this disease, this virus, this bacteria, you have to go through breeding and use the genomics to differentiate and pick up the families. And so we may be, let's say, 2 or 3 years behind, but with these technologies, I mean, you can easily step up in 6 months.
Well, maybe I just want to add a little bit. I think that when you compare, I think the main difference between Norway and Southern Europe is really organization. When they have a problem, they go for it. You know, of course, they have money, they have a backup from the government, and so they resolve the problem. Why have they not really applied this kind of technology? Because of a technical problem. The technical problem was mentioned, but not in detail, is that the salmon has an extra genome duplication. What this means is, instead of having like 1 or 2 copies of a gene, which is not identical, it's similar but not identical, they have twice as much. And this second replication happened recently. So that means that they have copies which are almost identical. So if you want to map, you don't know where to put it because, you know, is this the gene A or gene B, or is it gene B or gene A? But in able to apply this kind of technology like, you know, PPL and so on, you have to have a map which is precise and that you don't have doubts about what it is. So they, although they have the money, they have the organization, they come up with a technical problem, which they have not been able to put into practice. So what are they doing now? They are spending much more money to sequence even more. So they try to get really high quality sequences to be able to then, even things that are only slightly different, they can distinguish each other. So I think this is important to see. So in a way, we have recovered because they started earlier. We have recovered. We don't have kind of technology had made a big effort, and that's where we are. With a bit of luck in the middle, because I think there was some points where we were lucky. But I would say we are ready and probably more prepared than they are in some respects.
So they are. Sorry, Costas.
I may add that another advantage of the Norwegians is that they sell better.
They sell better.
Yeah.
The results, you mean?
The results, their effort, their whatever, and they collaborate with each other. Compared to us, the result.
They are punished.
So they have all these problems that said, but they sell better. You see, you said that you thought they're ages ago.
Ahead, yes. But this means that we are not aware of the developments that happen in the Mediterranean. So more and more should be transferred to people from the industry to talk about it, and then for me, Saropoulou, QTL, or whatever it was for Lokai, for NODA, it's extremely important. And this could be the base of a new proposal with a goal to put together something to do real work, industry work.
Yeah, but you also remember.
Nobody knew that. We don't read very, very sophisticated magazines.
Yeah, but it was, I remember your proposition for the ATIP, for the Greek branch of ATIP, I mean, to put in the intranet this kind of publications that might be interesting.
No, we can do that easily.
Yeah.
And we have to do that.
FGM, you know.
GFM. Any other questions? I think we should take this opportunity to think about consortium. Many companies could participate in a consortium to put together a proposal for this call, focused on NODA. It's an opportunity to just apply, but in the meantime, have your plan with your strategic plan of how you tackle the problem to discuss among the industry. So thank you, Costas.
As I saw in your mapping of wild and farm fish in Spain and in Greece, the Spain wild fish are about the same with the Greek farm, or not?
Yes.
So maybe it has to do, this one, yes. So, Spain wild, Spain farm and Greek.
It's for Seabass or Sea Bream?
Both of them.
Yeah.
They have some very common.
Scattered.
Yeah.
Scattered quantity of that one.
Yes.
So maybe that means that the broodstock of Greek farms are from Spain, as I understand.
No,
separate.
The wild Greek is this one. Wild Greek, wild Spain, farm Spain, farm Greek. Farm Greek is always apart. It has nothing to do
From another planet.
It's probably country.
In the winter, but know if they are spawning, if they have, if the eggs are fertile, and we have this kind of question.
Yeah, but you can imagine. I don't remember the percentages, but I have analyzed, let's say, more or less 300 eggs, and 80% was sea bream. But okay, it was close to the cage. It's not to the open sea.
Okay. Next.
Another question. As I understand, if you have a tank of broodstock of 200 fishes, with this technology, you don't need this. You need only 50 or 60 fishes. Because you
only a
You
few
can
groups.
find which species are spawning and keep only this species. Because somewhere you said that
Yes, but
only 30% or
yeah, but it's not always a question of the fish that participates now will going to participate afterwards, because there are some behavioral also reasons. If you put this 54 fish, let's say, if you put the 1/3 that participates in 200, this 70 fish, if you put them only these 70 fish, maybe again 1/3 of them is going to participate.
yeah.
But what happens is that day,
It's 1 day. It's not
Yeah,
a
that's
4
1 day.
months.
Tomorrow to be
Ah,
fish.
okay.
1 day.
1 day.
You take, you
Ah,
take
okay.
one spawning event.
Okay, I know that every day it's Sunday. Every day they are spawning only 2 or 3 fishes.
They don't change.
Yes, we don't know. We know it, but I
Yeah,
think it
but-
was
No,
4
it's
months
Bream, it's
cycle.
not like that.
Yeah.
See, Bream, most of the fishes, maybe more than 30%, 50%.
Costa, go to the next presentation.
But there is the next day, the other.
Yes, yes, okay. No, I thought it was in 4 months period, not in 1 day.
Okay.
And as I understand, this technology